Hello, and welcome to our microscopy and staining lecture. It's a easier unit module than cell structure and function, so this one was a little lighter to pair with it in the same week. So as you go through the cell and see how much content there is in the cell, it's important to realize that that's kind of building on a lot of things, and we even included some bacterial division in there. Um, so with this two sets of videos, I'm going to go through microscopy in the first set and staining in the second set. So going right into it, we know we want to be able to uh, do these objectives by the end of these videos. You should have already looked over this PowerPoint a little bit and clued you in on to what to expect here. I've kind of gone over the mnemonics for gram stain as well, the purple positive, pink negative with the N, but we will see a few more things. So by the end of these lectures, you're going to want to be able to compare types of microscopy according to the appearance of observed objects. You will describe the gram stain. You should be able to describe all the steps mentally. Primary stain, mordant, decolorizer, counter stain, visualize. Then we're going to identify the gram reaction of any picture or slide of any organism that you see. It's simple, there's only two choices, so you either see that purple or you see that pink. Finally, we want to be able to classify bacteria according to shape and staining characteristics. Okay, shape is bacillus or coccus, and we've kind of gone over those several times now. But for the staining part is in this lecture, and you'll want to see if it is purple or if it is pink, okay, or pink reddish or purple. And it'll be always a deep purple in the lab, so you shouldn't be able to question whether it's positive or negative. Trust me. All right, so as promised, our first part is going to be microscopy. Okay, and this is in part of microscopy and staining, and just like we see over here, we want to be able to tell that that is gram-negative bacillus with two flagellas at the end. Okay, so that's like a tuft of flagella. Those like Lophotrichus. Okay, and then we have a long chain of coccus. Okay, that would be streptococcus. Okay, so that's a gram-positive streptococcus and uh, a gram-negative bacillus with flagella and fimbriae. Remember, those are fimbriae, not um, cilia, because they are bacteria. All right, so that's kind of it in a nutshell that you'll be able to do by the end of this. But first, we're going to go ahead and look at the microscope. That's all the way over there. Okay, and remember, I've said that light is the most important part of a microscope, at least in the light microscope range. Okay, and so what does that mean exactly? It means that we have all this spectrum of radiation, okay? It's called the electromagnetic spectrum, okay? And it's different types of energy, different types of wavelengths, okay? You've probably heard of gamma rays, X-rays. We all hear the radio and have watched TV using these waves. These waves are the same waves as light. They're blasted out into space. They're going out the speed of light just like light is. Our shows that we started broadcasting in the 50s have a finite place that they are in the universe right now, and we know exactly where that is because we know how fast it's going. But the main thing is that we see the small range right here that looks like this wavelength of light. Okay, and all the purples, all the reds, and all the greens that we've seen are all nice and tucked into that little spectrum of light. Okay, so we just don't have receptors to see these. We don't have receptors to see these. Some animals have receptors to see UV, and some animals have receptors to see infrared, but we do not. And in fact, that's why we kind of name those infrared, meaning lower than red, and ultraviolet, meaning above violet. So we kind of name it based on uh, the human aspect anyway. All right, so remember, visible light is the main thing that you can get from this slide. Visible light is a narrow spectrum of the electromagnetic spectrum. Okay, and we use this light to come to a receptor in our eye to be able to see the image like so. Okay, so the light is going to refract upon the glass lenses, the objective lens, and then the ocular lens. And the idea is that we are going to take the specimen and enlarge it to a point that we can fix at a focal point, and then re-enlarge it back to where it hits the back of our retina, and we can contrast between it and the background. Okay, so contrast is really important in these clear cells that don't have any color. It's really hard to see things. That's also why we use the cotton ball and the algae and the dirt of the water, so you can kind of have some contrast without staining, and that way they'd stay alive. And then obviously staining can increase this contrast, but usually staining means you're going to have to kill whatever you stained in order to stain it. Okay, and so that's the trade-off. All right, and so when we use light microscopy on a bright field microscope, that is what we're doing in the lab. Okay, so this is the one that you kind of want to remember. Okay, and so we don't use a simple light field microscope. You could find those probably in a kid's store, but it's really just like a magnifying glass, and so it's just one lens, and so that's just kind of like a magnification. Okay, and Leeuwenhoek used his little type that you look through. That was a simple microscope. Okay, what we have in the lab is a compound microscope. All right, and it's a series of lenses for magnification. So there's some lenses in there that go to 40x, some lenses in there that go to 100x, and then there's the ocular lens. Okay, and then you'll multiply those to get our total magnification. Okay, and so now we've gone over the total magnification a few times as well, and so we understand that that's one of our importance. Okay, so remember, compound microscope, it's called compound because we compound the magnification. Okay, and oil immersion will increase our resolution. It will draw the light closer to our eye so we can determine it. All right, so we've gone over this a few times as well, and I will make a video of this going over the parts of me holding a microscope. I've already made the video, just gotta edit it a little bit. All right, so I'll go through this kind of quickly. All right, remember, light needs to go through the specimen, through the objective lenses, and then through the ocular lens. Okay, and then out to our eyes, and then that's how we are going to see it. So the light is the most important. Okay, the base and the arm are for storage and moving it. Okay, but that base contains the illuminator, the light source, and on the side we can kind of lessen and control that light. You have the condenser and the diaphragm. The condenser brings the light closer and further, and the diaphragm kind of closes the condenser and the light beneath the stage, so you can let less light through depending on how much you need. Okay, so that is our light. Once we have our light, we need our specimen on the stage, being held by stage clips. We will position it using the stage knobs. Okay, That goes left and right and front to back. And then we will look through it, through the objective lens and through the ocular lens in order to see that specimen. Okay, and then we will use the coarse focusing knob to move it up and down into plane to make it contrast on our retina, and we'll use the fine focusing knob so we can actually see the item. Okay, so that's the microscope. Real quick, use this, pause it at certain parts if you need to. You will be able to want to identify parts of the microscope on both exams and the lab quiz and the lab practical. Oil immersion, I kind of drew this on the board, but this is a better picture of it. All right, so as you can see in the picture on the left, you see that the light is scattering away from the objective lenses. All right, it's not touching the lens, which means your eye can't see it. It's touching the wall of the objective lens. Okay, but that immersion oil lowers the refraction and it makes more light enter the lens. Okay, so instead of refracted and lost, it concentrates it into a spot and now you can see it. So that's what we're getting with the oil immersion, and that's why I included it in that video. I'm probably going to edit it out, but you know, that's why we included it, because oil immersion is going to increase our contrast of things we stain, especially gram stains. Okay, and just so you can be brought up to speed, there's bright field microscopes, which we'll use in the lab. Also, types of light microscopy are dark field, Normarsky, and phase contrast. You're going to want to know dark field, even though we won't use it, so knowing bright field versus dark field is a good thing to know. Finally, electron microscopy, okay, is very important for us, because this is how we're going to see viruses. There's questions on how you can see viruses, and they only use the electron microscope. Okay, so light microscopes can't resolve down to a certain resolution, so electron microscopes can, and they give us back these beautiful 3D images. These are all colorized by either AI or an artist. The microscopes don't give back color. But you can have two different types, and you'll want to know the difference on how to tell them. If you see these nice 3D images, you see a scanning electron microscope. And if you see highly detailed but 2D images, you get a transmission electron microscope. The transmission one basically means that you can see through items, and you can see through items to see 2D detail. The scanning one scans these things and makes a nice 3D image. So that's kind of how you could tell them apart. And that'll conclude our microscopy portion, and we will be back with the staining portion.